How can we create self-sustaining mini-Earths that can grow and thrive in space as enclosed ecosystems with biodiversity? The idea of building mini-Earths or self-sustaining enclosed ecosystems in space is a fascinating interdisciplinary challenge that spans ecology, engineering, astrobiology, and space systems design. Although we are far from having a turnkey solution, here are some key considerations and research directions that scientists and engineers are exploring to move toward this ambitious goal. 1. Understanding closed ecosystems. Historical precedents. Experiments like NASA's Biosphere 2 and various closed-loop life support systems on the International Space Station ISS have taught us much about the challenges of sustaining life in a sealed environment. These projects highlight the complexity of balancing gas exchange, water recycling, waste management, and nutrient cycles. Key components. A fully self-sustaining ecosystem would need to mimic Earth's interdependent cycles. Atmospheric regulation, maintaining the right mix of oxygen, carbon dioxide, and trace gases. Water cycle, recycling water through condensation, filtration, and biological processes. Nutrient cycling, decomposition, soil fertility, and nutrient replenishment. Energy input, providing sufficient and controlled energy, e.g., sunlight or artificial lighting, to drive photosynthesis and other key processes. 2. Design considerations for space habitats. Structural design and materials. The habitat must be robust, airtight, and radiation shielded. Lightweight composite materials, advanced polymers, or even self-healing materials might be used to create structures that can withstand the rigors of space. Gravity simulation. Microgravity affects plant growth, fluid dynamics, and animal behavior. Rotating habitats or using centrifuge-based systems could help simulate gravity, which might be necessary for some ecosystem processes. Environmental control systems. Advanced sensors and control algorithms are essential to monitor and adjust environmental parameters, temperature, humidity, atmospheric composition in real time. This could involve AI-driven management systems that learn from the ecosystem's behavior and adjust parameters accordingly. Energy management. A reliable, renewable energy source is critical. Solar panels, possibly augmented by energy storage systems like batteries or fuel cells, would likely provide the necessary power for lighting, climate control, and other systems. 3. Integrating biodiversity. Plant life. A variety of plant species will be chosen not only for food production, but also for oxygen regeneration and CO2 absorption. Selecting plants that are resilient in lower gravity and confined spaces is essential. Microbial communities. Microorganisms play a pivotal role in nutrient recycling and waste decomposition. A controlled consortium of bacteria, fungi, and other microorganisms can help maintain soil health and support plant growth. Animal life. Small animals or insects could be introduced to pollinate plants, help decompose organic matter, or serve as part of a food chain. Their introduction must be carefully controlled to avoid destabilizing the ecosystem. Biodiversity and resilience. Just as on Earth, diversity can buffer the system against perturbations. The challenge is to design an ecosystem where the interactions are well understood enough to predict and manage the consequences of any imbalance. 4. Challenges specific to space. Radiation exposure. Space habitats must protect life from cosmic rays and solar radiation. This might involve using regolith, lunar or Martian soil as a shielding material, water walls, or other innovative radiation protection techniques. Isolation and autonomy. A self-sustaining mini-Earth in space would ideally operate independently for extended periods. This means that all biological and physical processes need to be highly reliable, and the system must be robust to failures. Scaling issues. The size of the ecosystem affects its stability. A very small system might be too fragile to absorb shocks, while a larger system is more robust but also more challenging to control and maintain. 5. Pathways toward realization. Incremental experimentation. Begin with small, modular experiments in controlled environments on Earth. Use these to test and refine closed-loop life support systems, simulating the conditions of space as closely as possible. Orbital demonstrations. Once small-scale experiments are successful, move to testing prototypes on the ISS or other orbital platforms. These experiments can validate models of closed ecosystems in actual space conditions, including microgravity and radiation exposure. Interdisciplinary collaboration. This challenge requires collaboration among ecologists, aerospace engineers, materials scientists, computer scientists for automation and control, and even ethicists. Establishing international research programs and test beds will accelerate progress. Adaptive and resilient design. Incorporate principles from systems biology and ecological engineering to design ecosystems that can adapt to changes and recover from disruptions. This may involve genetic engineering of plants and microbes for enhanced resilience, though such approaches would also require careful ethical and safety evaluations. 6. Ethical and practical considerations. Risk management. Any attempt to create a self-sustaining ecosystem, especially one that might be expanded or relocated, must consider potential risks such as system collapse or unintended ecological consequences. Robust containment and fail-safe systems are essential. Long-term monitoring. Even a well-designed ecosystem will require ongoing monitoring and possibly intervention. Planning for long-term management, including remote troubleshooting or even robotic assistance, is crucial. Regulatory and ethical oversight. As we push the boundaries of creating life-supporting habitats, international guidelines and ethical frameworks will be necessary to ensure that such technologies are developed and deployed responsibly. Creating self-sustaining mini-Earths in space is an immensely challenging but potentially transformative endeavor. It requires advanced materials and structural engineering to build safe, long-lasting habitats, deep ecological understanding to design and maintain balanced, closed ecosystems, innovative energy and environmental control systems that can operate reliably in the harsh conditions of space. Step-by-step experimental validation, starting with small-scale Earth-based models and moving to orbital prototypes. While significant scientific and engineering hurdles remain, research in areas such as regenerative life support systems, habitat design, and controlled ecological engineering is steadily paving the way toward this futuristic vision. How can we genetically engineer these self-sustaining habitats to adapt to extraterrestrial environments in space? Genetic engineering can play a pivotal role in adapting organisms for self-sustaining habitats in space. By tailoring biological systems to cope with extraterrestrial challenges, we can create resilient, efficient life support components for mini-Earths. Here are several strategies and considerations. 1. Enhancing stress resistance. Radiation tolerance. Space environments expose organisms to higher levels of cosmic radiation. We can transfer genes from extremophiles like Deinococcus radiodurans that have robust DNA repair mechanisms or antioxidant systems to protect critical cellular functions. Microgravity adaptations. Microgravity affects cellular processes, such as nutrient transport and growth. By modifying signaling pathways or structural proteins in plants and microbes, we can help them maintain normal physiological functions and structural integrity in reduced gravity. Temperature extremes and vacuum. Genes from extremophilic organisms can be introduced to enhance tolerance to temperature fluctuations and desiccation. For example, stress response genes that promote the production of protective proteins or molecules can be engineered to help organisms survive rapid environmental shifts. 2. Optimizing metabolic efficiency. Enhanced photosynthesis and carbon fixation. Genetic modifications can improve the efficiency of photosynthesis in plants. This might involve altering pigment composition or enzyme kinetics to better utilize the available light spectrum in space habitats, including adapting to artificial lighting conditions if necessary. Nutrient recycling. Engineering microbial communities to optimize nutrient cycling is essential. This includes modifying pathways for nitrogen fixation, phosphorus solubilization, and organic matter decomposition to ensure a continuous supply of nutrients in a closed ecosystem. Water use efficiency. Given the limited water supply in space, modifying plants to use water more efficiently through changes in stomatal regulation or root architecture can be critical for maintaining a balanced water cycle. 3. Facilitating symbiotic relationships. Plant-microbe interactions. Engineering plants to exude specific compounds that favor beneficial microbial colonization can enhance nutrient uptake and stress resistance. In turn, microbes can be designed to produce growth-promoting substances, antibiotics, or stress relief molecules. Integrated biosynthetic pathways. Synthetic biology allows us to design new biosynthetic pathways that facilitate symbiotic relationships, ensuring that the waste products of one organism become the resources for another. This is key to creating a self-sustaining cycle that mimics Earth's ecosystems. 4. Customizing organisms for extraterrestrial conditions. Tailored organism profiles. Not every organism from Earth is ideally suited for space. By editing genomes, we can create or enhance organisms that thrive under low atmospheric pressures, altered gas compositions, or even in habitats with artificial gravity. For example, modifying cell wall composition in plants could help them withstand variations in atmospheric pressure. Artificial selection and directed evolution. In tandem with genetic engineering, directed evolution techniques in controlled environments can help select for traits that perform best under simulated extraterrestrial conditions. This iterative process can fine-tune organisms for optimal performance in space habitats. 5. Ethical, safety, and regulatory considerations. Containment and monitoring. Any genetically engineered organism designed for space must include built-in safety measures, such as genetic kill switches or containment strategies, to prevent unintended ecological consequences. Ethical oversight. The engineering of organisms for extraterrestrial environments raises ethical questions about altering life forms and potential impacts on off-Earth environments. It is crucial to engage with ethicists, regulatory bodies, and international space agencies to establish robust oversight frameworks. Long-term viability. Ensuring that engineered organisms remain effective and safe over long periods in closed-loop systems is a major challenge. Continuous monitoring, along with the ability to update or replace genetic systems as needed, is essential for maintaining ecosystem balance. Genetic engineering offers a promising path to tailor organisms specifically for the demands of extraterrestrial habitats. By enhancing stress tolerance, optimizing metabolic pathways, and fostering symbiotic relationships, we can build resilient, self-sustaining ecosystems that mimic Earth's biodiversity, even in the harsh conditions of space. However, this endeavor requires careful design, rigorous testing, and comprehensive ethical and regulatory oversight to ensure success and safety in creating living, thriving habitats beyond our planet. Creating self-sustaining, closed-loop, productive systems that thrive in space involves an intricate integration of biology, engineering, and environmental control. The overall goal is to design an ecosystem in which every output is transformed into an input, mirroring the natural cycles found on Earth. Here is a detailed explanation of the processes involved. 1. Designing the structural framework. First, a robust and secure habitat must be engineered to house the ecosystem. The structure should be airtight and shielded against the harsh conditions of space, including cosmic radiation, extreme temperature fluctuations, and microgravity or variable gravity environments. Engineers must choose lightweight yet durable materials, such as advanced composites or radiation-resistant polymers, and design the habitat with redundancy in life support systems. Additionally, environmental control systems need to be integrated to maintain stable temperature, humidity, and atmospheric conditions inside the habitat. 2. Establishing closed-loop life support systems. A self-sustaining system relies on closed-loop life support, where all resources are continuously recycled. This involves atmospheric regulation. Specialized sensors and automated control systems monitor the balance of oxygen, carbon dioxide, and other trace gases. Plants and engineered microorganisms perform photosynthesis and respiration in cycles that keep the air composition stable. Water recycling. Water is an essential resource, so the system incorporates processes such as condensation, filtration, and purification to reclaim water from all sources, including human waste and transpiration from plants. Advanced water treatment technologies are integrated to ensure that water remains free of contaminants. Nutrient cycling. Waste products from humans, animals, and plants are broken down by microbial consortia into their basic components. These nutrients are then reabsorbed by plants, which, in turn, provide food and oxygen. This process mimics Earth's natural nutrient cycles and requires careful calibration to ensure balance. Energy management. A reliable energy source is critical. Solar panels, possibly supported by energy storage systems like batteries, power the habitat systems and any artificial lighting used for plant growth. The system must ensure a continuous energy supply to support all life support processes. 3. Integrating biodiversity. Biodiversity is a cornerstone of resilience and productivity in any ecosystem. In space habitats, careful selection and integration of various species is required. Plant life. Multiple plant species are selected for their ability to perform photosynthesis, provide food, and help stabilize the atmosphere. Plants are chosen based on their resilience to low gravity and confined spaces, as well as their nutritional profiles. Genetic engineering may be used to enhance traits such as drought tolerance, nutrient uptake, and radiation resistance. Microbial communities. Microbes are indispensable in decomposing waste and recycling nutrients. A consortium of bacteria, fungi, and other microorganisms is engineered or selected to ensure that all organic matter is efficiently converted into usable forms of nutrients for plants. These microbial communities may also help protect plants from pathogens and stress. Animal and insect life. In some systems, small animals or insects are included to help with pollination, organic matter breakdown, and maintaining balance within the ecosystem. Their roles are carefully controlled and monitored to avoid ecological imbalances. For optimizing metabolic processes through genetic engineering. Genetic engineering plays a critical role in adapting organisms to the unique challenges of space. Scientists work on modifying the genetic makeup of plants, microbes, and even small animals to improve their performance in closed-loop systems. Stress resistance. Genes that confer resistance to radiation, extreme temperatures, or desiccation are introduced. For instance, DNA repair genes from radiation-resistant bacteria such as Deinococcus radiodurans can help protect cellular structures against cosmic rays. Enhanced photosynthesis and nutrient uptake. Modifications to improve the efficiency of photosynthesis allow plants to maximize the conversion of light energy into chemical energy, even under artificial lighting or altered light spectra in space. Additionally, genetic changes may enhance root systems for better nutrient absorption in confined soil or hydroponic setups. Symbiotic relationships. Engineering plants to secrete compounds that attract beneficial microbes can further optimize nutrient cycling. Similarly, microbes can be modified to produce growth factors that enhance plant resilience and productivity, creating a tightly integrated system where every organism supports the others. 5. Developing and integrating automated control systems. Given the complexity of these ecosystems, automated control systems are essential for continuous monitoring and real-time adjustments. Sensors distributed throughout the habitat collect data on atmospheric composition, water quality, temperature, humidity, and plant health. This data is fed into a central control unit, often powered by AI algorithms, which adjusts environmental parameters to maintain balance. This system can autonomously handle routine fluctuations, detect anomalies, and alert operators to potential issues before they become critical. 6. Testing, iteration, and scalability. Before deploying a fully integrated system in space, extensive testing is conducted on Earth. Initial experiments are carried out in controlled laboratory settings, followed by small-scale prototypes that simulate space conditions. Incremental scaling allows engineers and scientists to address challenges such as system fragility in miniature ecosystems or the need for robust fail-safes in closed loops. Orbital demonstrations on platforms like the International Space Station help validate these designs under actual space conditions, including microgravity and increased radiation exposure. 7. Long-term management and ethical considerations. Finally, creating a self-sustaining system in space is not a one-time event but requires long-term management. Continuous monitoring ensures that the ecosystem adapts to evolving conditions and that any failures are swiftly corrected. Ethical considerations also play a vital role. Every genetically modified organism must include safeguards such as genetic kill switches to prevent unintended proliferation. Regulatory frameworks and international guidelines must be followed to ensure that these experiments are conducted responsibly and with respect for potential extraterrestrial environments. In summary, creating self-sustaining, closed-loop, productive systems in space is a multifaceted process that involves the design of a robust structural habitat, the integration of advanced recycling and nutrient cycling processes, and the careful selection and genetic engineering of organisms to thrive in harsh conditions. Automated environmental control systems and iterative testing ensure the stability and scalability of the ecosystem. With rigorous long-term management and ethical oversight, such systems can not only support human life but also pave the way for sustainable living in the broader context of space exploration and colonization. How can these closed-loop ecosystems support humans and other life forms in space? Closed-loop ecosystems are designed to recycle resources continuously, making them ideal for supporting humans and other life forms in space. Here's how these ecosystems function to meet the needs of their inhabitants. Oxygen production and carbon dioxide removal. Plants and photosynthetic microbes convert carbon dioxide into oxygen through the process of photosynthesis. This not only creates a breathable atmosphere for humans and animals, but also helps regulate CO2 levels. In a closed-loop system, the oxygen produced by plants is continuously replenished, ensuring a stable atmosphere. Water recycling and purification. Water is a precious resource in space, so these ecosystems are built to reclaim and purify water from all available sources. Techniques such as condensation, filtration, and microbial treatment transform wastewater, including human waste, plant transpiration, and other runoff, back into potable water. This constant recycling reduces the need for resupply from Earth and supports long-term habitation. Nutrient cycling and food production. In a closed-loop ecosystem, organic waste is broken down by microbial communities into essential nutrients that plants can absorb. This nutrient recycling creates a sustainable food chain. Plants not only supply oxygen but also provide fruits, vegetables, and other edible biomass, directly supporting human nutrition and potentially the diets of other life forms within the habitat. Waste management and energy flow. All biological waste products are managed within the system. Decomposing organisms convert waste into usable nutrients, which are then taken up by plants and other organisms. In turn, this supports the entire food web and maintains a balance of energy flow throughout the ecosystem. Such waste-to-resource conversion is critical in a space habitat where external inputs are limited. Integrated biosphere control systems. Automated sensors and control systems monitor environmental conditions, such as air quality, humidity, temperature, and nutrient levels, ensuring that the habitat remains stable. These systems make real-time adjustments to maintain equilibrium within the ecosystem, thereby protecting the health of humans and other organisms. By continuously optimizing conditions, the closed-loop ecosystem remains productive and safe for life. Adaptation to space conditions. Through genetic engineering and careful species selection, organisms in these ecosystems are optimized to withstand space-specific challenges like microgravity, higher radiation levels, and confined living conditions. Plants may be engineered for improved growth in low gravity, while microbes could be enhanced to break down waste more efficiently under these unique conditions. This tailored resilience is key to sustaining life in the extreme environment of space. Supporting diverse life forms. Beyond human survival, closed-loop ecosystems are designed to support a wide range of life forms. Diverse species, including plants, microbes, insects, and even small animals, contribute to ecosystem stability. For example, insects can aid in pollination, while a varied microbial community ensures efficient nutrient cycling. This biodiversity enhances the ecosystem's resilience against environmental fluctuations and potential system failures. In summary, closed-loop ecosystems support humans and other life forms in space by creating an interconnected, self-sustaining environment. They provide essential life support functions such as oxygen generation, water purification, nutrient recycling, and food production, while adapting to the unique challenges of space. Through integrated control systems and tailored biological components, these ecosystems offer a promising path for long-term, sustainable living beyond Earth. What species would be most suitable to grow, produce, and support human and other life forms in these systems? How will water circulate to nurture all life in these habitats? When designing closed-loop ecosystems for space, the choice of species and the water circulation system are both critical for ensuring long-term sustainability and resilience. Here's a detailed look at each. Species selection for a self-sustaining habitat. Plants. Plants form the backbone of these ecosystems, providing oxygen through photosynthesis, food, and even contributing to water purification through transpiration. Food crops. Species such as lettuce, spinach, tomatoes, and potatoes are excellent candidates because they have relatively fast growth cycles and provide essential nutrients. Grains like wheat or rice may also be integrated into the system, but they might require more space and time. Microgreens and herbs. These are ideal for quick yields and can be grown in compact spaces. They contribute to a varied diet and can also be used for medicinal purposes. Algae. Certain strains of algae, like spirulina, offer high protein content and are highly efficient at converting light into biomass, making them attractive for both nutritional value and oxygen generation. Microbial communities. Microorganisms play a vital role in recycling nutrients, decomposing organic waste, and even purifying water. Nitrogen-fixing bacteria. Bacteria such as Rhizobium or Azotobacter can convert atmospheric nitrogen into forms that plants can absorb, ensuring a steady nutrient supply. Decomposers. Fungi and bacteria are essential for breaking down organic matter, turning waste into nutrients that plants can reuse. Bioremediation microbes. Certain strains are capable of breaking down toxins or excess waste products, ensuring that water and soil remain healthy. Insects and small animals. While their role is more specialized, including small invertebrates can enhance the ecosystem's resilience. Pollinators. In a confined environment, small pollinators like certain species of bees or flies, or even engineered pollinators, may be introduced to assist with plant reproduction, though care must be taken to control their populations in a closed space. Decomposers. Some insects, like detritivores, can help in the breakdown of plant matter and animal waste, further contributing to nutrient cycling. Small vertebrates. In some designs, small animals such as certain fish species and aquatic components, or even lab-tested rodents may be part of a broader food chain, provided their care and waste management are well integrated into the system. Genetically engineered organisms. In many cases, genetic engineering may be employed to enhance the inherent resilience of these species. For example, plants may be modified to tolerate low gravity or high radiation, while microbes might be engineered for more efficient nutrient cycling or waste breakdown. These modifications can ensure that the selected species are well adapted to the unique challenges of a space environment. Water circulation in closed-loop habitats. Water is the lifeblood of any ecosystem, and in space habitats, its careful management is critical. The water cycle in these systems is designed to capture, purify, and redistribute water continuously. Collection and condensation. Transpiration. As plants photosynthesize, they release water vapor through transpiration. This vapor can be captured by condensation systems within the habitat. Wastewater recovery. Water from human and animal waste, along with gray water from cleaning and other processes, is collected in a centralized system. Purification and recycling. Filtration systems. Advanced filtration techniques, including mechanical filters, activated carbon filters, and even UV or ozone treatments, are used to remove contaminants and pathogens from the collected water. Biological purification. Beneficial microbes can be integrated into bioreactors that mimic natural wetland or soil processes. These systems help break down organic compounds and convert waste into nutrients while cleaning the water. Condensers and dehumidifiers. Water vapor is condensed back into liquid water using dehumidifiers and condensation panels, ensuring minimal water loss and efficient recycling. Distribution to ecosystem components. Piping and pumps. A network of pipes, pumps, and valves distributes purified water to various parts of the habitat. Automated control systems ensure that each section, whether plant growth areas, animal habitats, or human living quarters, receives the appropriate amount of water. Nutrient solutions. For systems like hydroponics or aeroponics, water is enriched with essential nutrients before being circulated to the plants, ensuring they receive balanced nutrition in addition to hydration. Feedback systems. Sensors continuously monitor water quality, pH, mineral content, contaminant levels, and flow rates. This data is fed into a central control unit that adjusts the water distribution in real time, ensuring that the ecosystem remains stable and responsive to changing conditions. In these closed-loop ecosystems, carefully selected species, from nutrient-rich plants and resilient microbes to supportive insects and small animals, work together to create a self-sustaining cycle that supports human life in space. Water, being continuously recycled through a combination of physical, chemical, and biological processes, is redistributed to nurture every component of the habitat. This holistic approach ensures that every element of the ecosystem reinforces the other, providing a stable, productive environment even in the challenging conditions of space. Boosting biodiversity, regeneration, and production rate in closed-loop space habitats requires a holistic approach that integrates advanced biological engineering, optimized ecosystem design, and real-time adaptive management. Below is a detailed breakdown of how to achieve these goals. 1. Enhancing biodiversity in space habitats. Biodiversity increases the resilience of an ecosystem by enabling complex interactions that improve stability, efficiency, and adaptability. In space habitats, where environmental fluctuations and resource limitations exist, diversity helps maintain balance. Strategies for increasing biodiversity. A species selection and genetic engineering. Polycrop systems. Instead of monocultures, multiple species with complementary functions should be grown together. For example, companion planting, e.g., legumes fixing nitrogen for grain crops, creates a natural synergy. Microbial consortia. A diverse mix of bacteria, fungi, and archaea enhances nutrient cycling, decomposition, and disease resistance. Genetic engineering. Enhancing species' ability to survive in space conditions, e.g., radiation-resistant crops, fast-growing plants, drought-resistant root structures, helps maintain biodiversity without excessive environmental control. Synthetic biology. Engineered microorganisms can be designed to support plant and animal life by improving waste breakdown, producing growth-enhancing compounds, or protecting against pathogens. B. Multi-trophic ecosystems. Food web balance. Pollinators. Introducing bees, flies, or robotic pollinators ensures successful plant reproduction. Detritivores and decomposers, including small decomposing organisms such as earthworms, fungi, and certain insect species, accelerates organic matter breakdown. Microalgae and cyanobacteria. These photosynthetic microorganisms contribute oxygen production and food for other species, improving ecosystem balance. Aquaponics integration. Fish and aquatic plants can be incorporated, creating a nutrient cycle between fish waste and plant growth. C. Ecological niches and redundancy. Each functional role in the system should have multiple species filling it. If one species struggles, another should be able to perform the same function. For example, multiple nitrogen-fixing bacteria species ensure nutrient cycling in case of environmental shifts. Various pollinators, bees, hoverflies, and engineered substitutes safeguard reproduction. 2. Boosting regeneration and resource cycling efficiency. Regeneration is crucial for sustainability in a closed-loop system. It ensures that resources such as oxygen, nutrients, and organic matter are continuously replenished. A. Optimized nutrient recycling. Microbial bioreactors. Engineered microbes break down organic waste efficiently, accelerating the return of nutrients to plant systems. Mycorrhizal fungi. These fungi extend plant root networks, improving water and nutrient uptake. Soil-less agriculture. Hydroponic and aeroponic systems eliminate soil degradation concerns, ensuring maximum nutrient efficiency. B. Advanced water reclamation. Fog capture and condensation. Water vapor from plants and human respiration can be collected and recycled. Biofiltration wetlands. Small-scale artificial wetlands with aquatic plants and microbes can filter water naturally before recirculating it. C. Enhancing photosynthesis and oxygen production. Algae bioreactors. High-yield algae such as spirulina can provide oxygen, sequester CO2, and act as a protein source. Multi-layered crop growth. Utilizing vertical farming techniques maximizes light use efficiency and produces more oxygen per square meter. 3. Accelerating production rates. Faster production of biomass, food, and oxygen is essential for sustaining a growing space habitat population. A. Improving plant growth conditions. Optimized light spectra. LED systems with adjustable blue and red wavelengths enhance photosynthesis efficiency. Artificial gravity, rotation-based systems, if available, artificial gravity through rotating habitats improves plant development and water distribution in root systems. Carbon dioxide enrichment. Higher CO2 levels within safe limits can increase photosynthesis rates and accelerate plant growth. B. Faster reproduction and growth cycles. Selective breeding. Over multiple generations, species can be selected for higher growth rates and better adaptability. CRISPR-based genetic enhancements. Modifying plants and microbes for rapid development, improved disease resistance, and higher nutrient efficiency. High-yield algae production. Rapidly reproducing algae provide continuous biomass output with minimal space and resource consumption. C. Artificial intelligence and automation. Automated climate control. AI-driven sensors adjust humidity, temperature, and CO2 levels in real time. Machine learning-based plant growth optimization. AI monitors plant health and automatically optimizes growing conditions to maximize yield. Robotic farming assistance. Robots handle pollination, pruning, and harvesting to accelerate production cycles. To maximize biodiversity, regeneration, and production rates in a closed-loop ecosystem for space, a combination of natural ecological balance and technological optimization is required. By diversifying species selection, integrating multi-trophic food webs, enhancing microbial activity, using genetic engineering, and leveraging AI-driven automation, we can create a highly resilient and productive space habitat capable of supporting human life indefinitely. How genetic engineering can be used to create the best species to make thriving, closed-loop ecosystems in space. Genetic engineering can enhance the adaptability, resilience, and efficiency of species used in closed-loop ecosystems in space. By modifying plants, microbes, and even small animals, we can optimize their functions for the unique challenges of extraterrestrial environments, such as microgravity, radiation, and limited resources. Below is a detailed breakdown of how genetic engineering can create ideal species for self-sustaining space habitats. 1. Genetic engineering for plants. Plants are the foundation of any closed-loop system, providing oxygen, food, water purification, and carbon dioxide regulation. Genetic modifications can enhance their ability to thrive in space conditions. A. Enhancing photosynthesis efficiency. Improved light utilization. Modify chlorophyll structures to absorb a broader spectrum of light, optimizing photosynthesis under artificial LED lighting. Introduce genes from deep-sea or shade-tolerant plants that enhance photosynthesis in low-light environments. Increase CO2 uptake. Enhance carbon fixation pathways, e.g., C4 or CAM photosynthesis in non-native species, to improve oxygen production. B. Accelerating growth and yield. Faster maturation rates. Modify growth-regulating genes, such as those controlling flowering and fruiting, to speed up plant development and increase food production. Dwarf varieties for space farming. Introduce genes that control plant height while maintaining high yields, optimizing growth in confined habitats. Increase nutritional value. Biofortify crops by adding genes that boost vitamin, mineral, and protein content. C. Stress resistance and adaptation. Radiation tolerance. Introduce radiation-resistant DNA repair enzymes from extremophiles, e.g., Deinococcus radiodurans. Microgravity adaptation. Modify root growth patterns to enhance nutrient uptake and reduce gravity. Engineer plants with increased resistance to water stress, ensuring survival in hydroponic and aeroponic systems. D. Waste recycling and nutrient cycling. Efficient nutrient uptake. Enhance nitrogen-fixing capabilities by introducing symbiotic bacterial genes into non-leguminous crops. Decomposition acceleration. Modify plants to produce enzymes that break down organic waste, accelerating composting and nutrient recovery. 2. Genetic engineering for microbes. Microbes are essential for nutrient cycling, waste decomposition, and environmental stability in a closed-loop ecosystem. Engineering them can improve their efficiency in maintaining ecosystem balance. A. Waste breakdown and nutrient recycling. Engineered decomposers. Modify bacteria and fungi to accelerate organic waste breakdown into usable nutrients. Introduce genes from lignin-degrading fungi, Trichoderma species, to enhance composting speed. Enhanced nitrogen fixation. Modify Rhizobium and Azotobacter to fix nitrogen more efficiently in various plant species. B. Water purification and oxygen generation. Bioluminescent microbes for monitoring water quality. Engineer bacteria that glow in response to toxic contaminants, acting as real-time biosensors. Algae and cyanobacteria for oxygen and water filtration. Enhance spirulina or chlorella with genes that increase oxygen production while also absorbing excess CO2 and nitrogen waste. C. Radiation and extreme condition resistance. Space-adapted microbes. Introduce radiation-resistant DNA repair systems into beneficial microbes to ensure their survival in space. Stress-resistant fermentation organisms. Engineer yeast and bacteria to tolerate high radiation while producing essential amino acids, vitamins, and fermented foods. 3. Genetic engineering for insects and small animals. Small animals and insects can play critical roles in pollination, waste decomposition, and protein production. A. Space-adapted pollinators. Resilient bee species. Modify bees to withstand space conditions, altering their circadian rhythms for artificial lighting. Introduce antifungal resistance genes to prevent colony collapse in confined environments. Robotic or engineered pollination alternatives. Modify hoverflies or ants to assist with pollination in enclosed spaces where traditional bees may struggle. B. Protein and nutrient sources. High-yield insects. Engineer mealworms or crickets with faster reproductive cycles and higher protein/fat content. Edible algae and fungi production. Modify fungi like Aspergillus oryzae to produce additional amino acids and vitamins for human consumption. C. Waste decomposition and soil enrichment. Super decomposers. Enhance Icenia fetida, red wiggler worms, to break down organic waste faster. Bioluminescent markers for ecosystem monitoring. Engineer insects with bioluminescence genes that activate in response to environmental stress, e.g., oxygen deficiency or toxic buildup. 4. Synthetic biology and artificial ecosystem engineering. Beyond modifying existing species, synthetic biology allows for the creation of entirely new organisms tailored for space environments. A. Custom microbial consortia. Artificially designed microbiomes. Engineer communities of bacteria and fungi that interact in a controlled way, ensuring stability in extreme environments. Bioengineered soil microbes. Develop microorganisms that extract and process essential minerals from lunar or Martian regolith, making extraterrestrial soil viable for plant growth. B. Self-healing and responsive ecosystems. Bioengineered biofilms. Create protective bacterial films that self-repair in response to environmental damage, sealing leaks or purifying air. Adaptive growth algorithms in plants. Engineer plants that dynamically adjust their growth rates based on resource availability using gene regulation sensors. C. Smart biosensors and environmental monitors. Bacteria with genetic memory. Engineer microbes that change color or emit signals in response to pollution or radiation levels, providing early warnings for habitat issues. Living biointerfaces. Develop plants that can serve as living sensors, changing leaf color in response to changes in soil nutrients or air quality. Genetic engineering holds immense potential for designing the ideal species for closed-loop space ecosystems. By enhancing photosynthesis efficiency, increasing growth rates, boosting resistance to space conditions, and optimizing microbial and insect contributions to nutrient cycles, we can create highly efficient, self-sustaining habitats through synthetic biology. We can go even further, developing entirely new life forms that process waste, generate oxygen, and recycle nutrients in ways that natural evolution has never produced. With these advancements, space colonies could sustain human life indefinitely, transforming extraterrestrial environments into thriving ecosystems. How can the aliens genetically engineer these to use them for different purposes? If an advanced alien civilization were to genetically engineer closed-loop ecosystems for different purposes, their approach could be vastly more sophisticated than ours, potentially integrating high-level bioengineering, nanotechnology, AI-driven evolution, and synthetic biology. These modifications could allow their ecosystems to function in extreme environments, serve as biomachines, or even act as intelligent life support systems. Here's a breakdown of how they might achieve this. 1. Purpose-driven genetic engineering of ecosystems. Aliens could design bioengineered habitats for a variety of specialized applications, ranging from self-sustaining colonies to bioweapons, terraforming tools, or even intelligent organisms capable of planetary-scale resource management. 8. Terraforming and planetary engineering ecosystems. Purpose: convert barren planets or moons into habitable environments by altering the atmosphere, soil, and water content. Genetically enhanced photosynthetic life. Engineered microbial mats, capable of surviving extreme temperatures and pressures, converting CO2 into oxygen more efficiently than terrestrial plants. Cyanobacteria that produce greenhouse gases to warm cold planets or reduce CO2 to cool hot planets. Self-replicating ecosystems. Alien-designed fungi and bacteria that break down rock into soil, making planetary surfaces fertile. Bioengineered extremophiles that convert toxic elements into usable nutrients. Be energy-producing biological systems. Purpose: create life forms that function as biological power plants. Photosynthetic energy farms. Alien-designed plants with direct energy conversion, absorbing light and producing electricity via bioengineered chloroplasts with nanomaterials. Microbes that convert waste into biofuel more efficiently than Earth-based anaerobic bacteria. Thermophilic energy organisms. Deep-sea or lava-dwelling species engineered to extract energy from volcanic vents or extreme heat sources. See sentient or semi-sentient ecosystems. Purpose: create biological systems that are self-aware, capable of autonomous adaptation and decision-making. Smart biocolonies. Alien ecosystems that contain genetically modified plants and fungi with networked neurotransmitters capable of environmental sensing and response. Trees or vines with bioelectric intelligence, capable of detecting and communicating changes in air quality, soil moisture, or radiation levels. Artificial intelligence embedded in biology. Alien-developed microorganisms that can act as biological processors, analyzing environmental data and making real-time decisions about resource management. 2. Adaptive bioorganisms for space travel. Aliens might design organisms that can live, function, and even move between planets or survive in deep space. A. Living spaceships, biocraft. Purpose: create ships made from biological materials that self-repair, self-adapt, and grow in space. Self-healing spacecraft. Alien species may have engineered organisms that serve as structural components for living spaceships. These ships could repair themselves from damage using regenerating cells. Symbiotic crew environments. Space habitats composed of bioengineered life forms that provide oxygen, food, and even emotional feedback through symbiotic interactions with their crew. B. Space surviving organisms. Purpose: create species that can survive in vacuum, high radiation, and extreme cold or heat. Radiation-absorbing microbes. Microorganisms that convert radiation into usable energy, protecting life forms within the habitat. Cryogenic suspension creatures. Alien organisms genetically engineered to enter deep hibernation, capable of restarting biological activity upon reaching a new planet or star system. 3. Biological weapons or defense systems. If an alien species were militaristic, they could design genetic organisms that function as weapons, defenses, or surveillance. A. Adaptive bioweapons. Purpose: organisms designed for targeted destruction of enemy life forms or ecosystems. Self-replicating plague organisms. Engineered viruses or bacteria that only attack specific DNA sequences, leaving their own species unharmed while devastating others. Bioterrain modification warfare. Deploying fungi or microbes that consume an enemy's crops or terraform their planet to an uninhabitable state. B. Living defense mechanisms. Purpose: defend against threats using engineered life forms. Biological sentinels. Alien-designed stationary bioweapons, such as trees that excrete deadly toxins or living barriers that react to intruders. Self-reproducing guard animals. Creatures genetically optimized for detecting enemies and protecting alien settlements without the need for traditional weapons. 4. Advanced life support systems and biological augmentations. Aliens could use genetic engineering to enhance their own biology or integrate biological systems directly into technology. A. Human-like symbiotic systems. Purpose: transform traditional life support systems into fully organic systems. Oxygen-producing skin. Alien species may have modified themselves to integrate algae or bacteria that produce oxygen directly into their bodies, reducing their need for artificial respiration systems. Living biofilters. Genetically engineered gut bacteria that detoxify poisons, remove radiation, and eliminate the need for waste management systems in space. B. Bioaugmented cyborgs. Purpose: merge biology with technology for enhanced capabilities. Genetically engineered neural interfaces. Direct brain-to-brain communication using living neurotransmitter networks. Regenerative healing systems. Self-repairing tissues infused with alien bionanotechnology that can regenerate lost limbs or organs in hours instead of weeks. 5. Multidimensional and quantum biology. If aliens have mastered higher dimensional physics, they could engineer life that interacts with dimensions beyond our understanding. 8. Phase-shifting organisms. Purpose: life forms that can exist between multiple states of matter. Quantum-adapted microbes. Organisms that exist in multiple locations simultaneously, allowing entire ecosystems to be distributed across planets without physical transport. B. Time-resilient species. Purpose: organisms that can endure time dilation effects in space. Biological stasis fields. Aliens may engineer time-locked species capable of surviving thousands of years in suspended animation, only reactivating when conditions become favorable. An advanced alien civilization could use genetic engineering to create fully autonomous, self-sustaining, and highly specialized ecosystems that serve as terraformers, energy producers, space travel support systems, biological weapons, and even intelligence networks. These modifications could extend into quantum biology, sentient life support systems, and planet-modifying organisms, allowing them to manipulate life in ways we can barely imagine. By designing ecosystems tailored to specific planetary environments, adaptive intelligence, and deep space survival, aliens could create an interconnected web of living technology capable of thriving across the cosmos.